Hey future doctors, today we are going to see about cell wall appendages. In part 1 we saw morphology of bacteria. So here the cell wall appendages. First is capsule and slime layer. So some bacteria possesses a layer of amorphous viscid material which lies outside the cell wall. So this is plasma membrane, this is cell wall, and again one more membrane which lies outside the cell wall, that is known as glycocalyx, which is made up of amorphous viscid material. Now this glycocalyx layer, if it is well organized and it is not easily washed off, then it is known as capsule. But if it is loose material and it can be easily removed, then it is known as slime layer. So here as you can see that this is a cell wall and this is a well organized glycocalyx. So this is capsule, and here also you can see this is a cell wall, then there is a glycocalyx layer which is loosely arranged, it is known as slime layer. Next point is some bacteria it possesses both capsule and slime layer. The example is Streptococcus salivarius. So additional to this, most of the bacterial capsules are polysaccharide in nature, but in Bacillus anthracis, the bacterial capsule is polypeptide in nature. Now there are some functions or uses of capsule. First, it contributes to the bacterial virulence. The capsule protects the bacterium from the phagocytosis. It also prevents complement-mediated bacterial cell lysis. It also prevents cell from drying out. It protects bacterium from action of lysosome and bacteriophages, as well as capsule of the certain bacteria such as Bacteroides fragilis. It may be toxic to the host cell, which induces the abscess formation. Then one more important point, that is the biofilm formation and adhesion. So biofilm is a living ecosystem which is made up of millions of adherent bacterial cells which are embedded within a self-produced matrix of the extracellular polymeric substances, that is known as polysaccharide slime layer. So here as you can see that this is a plastic surface, and on this plastic surface a biofilm is formed. It is a living ecosystem which is made up of various bacterial cells. If this biofilm is persistent, then it contains the pathogenic bacteria which are capable to adhere the damaged tissue as well as plastic surfaces such as medical devices such as catheters. So this is the first step of bacterial colonization and sometimes it leads to disease. For example, catheter associated urinary tract infection. So this was all about capsule and slime layer. Now next is flagella. So it is a thread-like appendages which protrude from the cell wall that confers the motility to the bacteria. It also acts as an organ of locomotion. Now on the basis of arrangement of flagella, it is divided into four types: monotrichous, lophotrichous, peritrichous and amphitrichous. As you can see in this figure, the monotrichous has the single polar flagellum, and examples are Vibrio cholera, Pseudomonas and Campylobacter. Then in lophotrichous, from a single pole, there are multiple polar flagellums. Example is Spirillum. The next is peritrichous. In this, the flagella is distributed over entire cell surface. And here the example is E. coli and Salmonella typhi. And the last is Amphitrichous, which has a single flagellum at both at its end. Here the example is Alcaligenes fecalis. So this was all about arrangement of flagella. So the ultrastructure of flagella, it has filament, basal body and hook. So filament is the longest portion of flagellum. It extends from the cell surface to the tip of the hollow rigid cylinder which is made up of single protein, that is flagellin. The next is basal body. So it is a portion of flagellum which is embedded in the cell. It is the most complex part of flagellum which is made up of 2 to 4 rings which is connected to the central rod. And the last is hook, which is a short, curved, flexible segment that links the filament to its basal body. So this is basal body, this is hook, and this is filament. This is all about ultrastructure of flagella. Now bacterial motility. So bacteria can produce characteristic type of motility which helps in their identification. So there are various type of motility and these are examples. First is tumbling motility. It is seen in Listeria. Then gliding motility. It is seen in Mycoplasma. Then stately motility in the Clostridium. Then darting motility in Vibrio cholera and Campylobacter. Then swarming on the agar plate. Examples are Proteus and Clostridium tetani. And last is corkscrew or lashing flexion extension modality, and here example is Paragit. So this was all about flagella. Now next is fimbriae or pili. So these are short, fine hair-like appendages which help in the bacterial adhesion, and these are known as organ of adhesion. The flagella was known as organ of locomotion, but pili or fimbriae, they are known as organ of adhesion. So the special type of pilus, that is known as sex pilus, it also exists which helps in the conjugation. So pili, they are made up of protein that is known as pilin, antigenic. Then they are not related to motility and it can found in both motile as well as non-motile organisms. Now there are various type of piles. So according to function, there are two type of piles: common pili or fimbriae, and next is sex pili. Common pili or fimbriae. These help in bacterial adhesion to the epithelial surface, and this is very small in size. The single bacterium can have as many of 1000 fimbriae. It is also present in gram-negative and some gram-positive bacteria. The next is sex pili. This help in bacterial conjugation by forming conjugation tube through which the bacterial gene transfer takes place. Pili are the long, thick tubular structures, and the number of sex pili are less, that is just 1 to 10 per bacterial cell. These are only found in the gram-negative bacteria. So these are type of piles. Now detection of fimbriae. So fimbriae can be detected either directly by the E microscope or indirectly through the formation of surface pedicle. As you can see, this is a surface pedicle which is formed. These are bacteria, aerobic bacteria, and this is a thin layer which is formed at the surface of liquid culture of the strongly aerobic bacteria such as Pseudomonas, which adheres to each other by their fimbriae. So next is spores, highly resistant, which is present in the dormant stage of the bacteria, and they are formed in the unfavorable environmental conditions. The result of depletion of nutrients. So let's discuss about the structure of spores. So from the innermost towards the outermost layer, it is core, then cortex, then coat, and last is exosporium. So the core, it is the innermost part which contains the DNA and is walled off from the cortex by the inner membrane and germ cell wall. Now cortex and the coat layer, they lie external to the core and are separated from each other by the outer membrane. Then the outermost layer is exosporium. So this was all about structure of spore. Now we are going to see about sporulation. So it is a process of formation of spore from the vegetative stage of bacteria. So it occurs due to the lack of nutrients. It is a very complex process, requires 10 hours. The mature spore formed is extremely resistant to the heat as well as disinfectant, which is due to deposition of calcium and dipicolinic acid into the spore cortex. The next process is germination. It is the transformation of dormant spores into the active vegetative cell when grown in a nutrient-rich medium. So if the spore got nutrient, it will germinate. Now let's discuss about the shape and position of spore. So there are different positions: central, subterminal, and terminal. Then on the shape, that is divided into oval or spherical. Then on the basis of if the diameter is less than the width of the bacteria, then it is known as non-bulging spore. Example is bacillus. But if the diameter of spore is more than the width, then it is known as bulging spore, and example is clostridium. As you can see in this figure, this is a non-bulging oval and terminal spore. Then second is non-bulging round and subterminal spore. Then third is non-bulging oval and central spore. But these three are bulging spores. This is a bulging round and terminal spore. Then this is a bulging oval and terminal spore. And this is bulging oval and central spore. Now next we are going to see about sporicidal agents. So what do you mean by sterilants? So these are capable of killing spores. Example of autoclave, ethylene oxide sterilizers. In demonstration, we can see at the gram staining, the spores appear as the unstained refractable bodies within the cell, but in modified Xenon staining, the spores are weakly acid fast and appear red color. As well as in the special techniques for the endospores such as Kaffir, Fulton stain and Moeller stain. Now we are going to see about application. So spores of the Geobacillus stereothermophilus, this is used as the sterilization control for autoclave and plasma sterilizer. And spores of the Bacillus atrophius, it is used as a control for air oven and ethylene oxide sterilizer. So this was all about cell wall appendages.